I am now going to lecture to the functional groups and linkages PowerPoint that you can find in the Unit 1 Notes folder. Okay, you can see on the first page there's a link to Crash Course Biology video. I'm not going to show that to you now. You can do that on your own by either clicking here or looking under the Unit 1 videos folder for it. It's the one on carbon. So we start off by talking about why carbon? Why is carbon seem to be the molecule or the atom, that is most important in organic molecules? Reasons for that are because it forms stable bonds with other carbon atoms. You can get long chains of carbon atoms or rings of carbon atoms. You can see a structure here with the rings of carbon attached to each other. These black spheres represent carbon atoms bonded to each other. It is the best electron share. It has a bonding capacity of 4, which means it can form bonds with up to 4 other atoms. And those bonds are covalent and they're quite strong. And those bonds also hold a lot of energy. So for those reasons, carbon is the most important atom in terms of life. Next, we move to discussing hydrocarbons. So hydrocarbons are compounds that are composed of hydrogen and carbon together. So they're combinations of hydrogen and carbon. And when you combine these two atoms and form long chains of carbon and hydrogen, these are non-polar molecules. And they store a lot of energy. But in order to be useful for living things, these hydrocarbons need to be able to interact in a watery environment. And that can only be achieved if a functional group that's polar is added to the molecule. So if you look at the molecule down here of stearic acid, which is an example of a fatty acid, most of the molecule is made up of hydrocarbon. So if it was just this material here, then it would be completely nonpolar. And we know that nonpolar compounds cannot dissolve in water. But by adding this functional group here, even though the overall molecule is still considered a nonpolar molecule, now we have a polar functional group added at the end. You can see that it's a polar functional group because it contains oxygen, which is highly electronegative. So by doing that, you can get this compound to interact with other compounds or other molecules in a watery environment. There's the highlighted carboxyl group, which is a polar functional group. So that was a carboxyl group. There are other functional groups as well, but you can see that most, if not all, of the functional groups contain an electronegative atom. So it makes them polar. So a hydroxyl group is an OH. There's your carboxyl group. An amino group, NH2. Sulfhydryl group is SH. Phosphate group is PO4. And then you have two types of carbonyl groups. A carbonyl group is when you have a double bonded oxygen to a carbon. If it's on a terminal carbon, meaning at the end of a chain, then it's considered an aldehyde. If it is on a central carbon, then it's considered a ketone. So those are both types of carbonyl groups. Now we'll look at two different types of reactions that occur between organic molecules. So these are two types of reactions. The first type is a condensation reaction. It's also called a dehydration synthesis. That's when you're taking two smaller molecules, taking their functional groups, interacting the functional groups to create a covalent bond and create a linkage. So you're basically taking two smaller molecules and creating a bigger one. So energy is required in this process. And it's also called an anabolic reaction because you're making a larger molecule. So when you think anabolic, think bigger molecule. So in this example here, we have two amino acids. So this blue thing and the green thing are both amino acids, separate amino acids. You can see that this amino acid has an amino group and a carboxyl group. And so does this amino acid, carboxyl group and amino group. If we position the amino group and carboxyl group of the adjacent amino acids close to each other, then the OH of the carboxyl group and a hydrogen of the amino group can be removed in the form of water, and you create a new covalent bond here between the two amino acids, and now the two amino acids are attached to each other. Notice that it removes water. That is why it's called a condensation reaction. When you think condensation, think removal of water. That's also why it's called a dehydration synthesis. D means to remove, so dehydration, removing water. So that's a condensation reaction. A lot of the molecules that we're going to look at later on in this unit, in this chapter, are considered polymers. And that's because they're long molecules composed of subunits. Those subunits are called monomers. So for example, the DNA molecule is made up of a long chain of nucleotides attached to each other. So the nucleotide is the monomer of the DNA, which is the polymer. And when all these monomers are attached to each other, it's using those condensation reactions that we saw on the previous slide. The other type of reaction we're going to look at is a hydrolysis reaction. This is basically the opposite of the reaction we saw with the condensation. So it's also called a hydration reaction. And in this case, if you look at the example, we have sucrose molecule, which is really made up of a glucose and a fructose attached to each other right here. And in this case, water is required. Water is not produced, water is required. So you can see the water coming in. And we've highlighted the water molecule here. The OH of the water molecule, when this bond is broken, gets added here, and the remaining hydrogen gets added to the oxygen here to create this. So basically, you're creating two new hydroxyl groups, and you're separating the sucrose into its component glucose and fructose molecule. So this is a process that releases energy. When these bonds are broken, energy is released. So it's also called a catabolic reaction, where you're taking something larger and breaking it into something smaller. Now, does that mean if I take sucrose, which is basically table sugar, and just add it to water, that I'm going to get glucose and fructose produced? No, that's not the case. When you add sucrose to water, as you know, when you add table sugar to water, it will just dissolve. It's still sucrose. So really, you need a catalyst for this reaction to occur. And a biological catalyst is an enzyme. And sucrase is the particular enzyme that's involved in causing this reaction to occur. Without it, the reaction would not occur, even if there's water around. These are some common biological linkages that I need you to know. When you have compounds that have a hydroxyl group, so you can see a compound here. Now the R just represents any hydrocarbon chain or ring. So if you have a hydrocarbon that has an OH group and another hydrocarbon that has an OH group, those hydroxyl groups can interact to produce an ether linkage. And you can see the arrow going both ways because when you create the ether linkage, water is removed. When the ether linkage is broken, water is added. If you take a hydroxyl group and a carboxyl group, you can see the interaction here. Water is removed. You create an ester linkage. When you take an amino group and a carboxyl group, you create an amide linkage. When you take a phosphate group and a hydroxyl group, you create a phosphate ester linkage. Now that's the end of the PowerPoint lesson, a short one today. What I suggest you do next is if you go to your home page, this should be what it looks like right now. If you go to content, you can see the note we just did was here. The textbook reference for this is still in this chapter 1.1 document. If you go to Unit 1 worksheets, I want you to open up the student note. Oops, that's the wrong one. I want you to open up the functional groups and linkages student note. And when you do that, you'll see this. It appears I'm running out of time for this video, so I'll continue this discussion in part 2.